我实验室做上面intern，那后来就是想要继续做influenza的实验，那就是有手上的潘建仁老师共同指导，但是实验是在中国这边做。那就是因为我们实验室主要是做influenza比较后期，那在plasma membrane的部分，但是其实这方面相关的有关 host 的资讯是比较少的，所以我们就想了一个方法，想要多找一些就是可以特定位置细胞里的位置跟感染时间点的proteomics方法，想要来多获取这边的资讯。那接下来就由核心科学的老师说明。
Hi,大家，我是郭鑫。So, just like Dr. Wang just introduced, today I'm going to talk about how we utilize the proximity labeling proteomics to explore the spatial temporal specific influenza virus host interaction in the virion formation at the plasma membrane. And in this feature, we established 3 different stable cell lines and used them to obtain the proximity labeling proteomics in order to investigate the potential host vectors that works in the influenza A virus. And so for starter, the virus was used in the study is the influenza A virus, and influenza A virus, also known as IAV, is a member of the Orsomyox viridae family. And IAV is a type of virus with negative sense single-stranded RNA genome which is segmented. And after IAV enters and replicated in the host cell, instead of forming a complete virion and release the complete virion directly out of the cell, the newly synthesized virion components will be recruited to the plasma membrane for the further assembling and budding and releasing process in the late stage. And therefore, this is the late stage of IAV infection that determines whether the contagious progeny virions are produced and released to infect the uninfected cells. And so the first-line treatment for influenza A virus mostly focus on this stage. So to better understand the host vectors that participates in this process, there are 2 major approaches that are used by the previous study. And so in the previous study, researchers mostly utilized 2 methods, which is the gene perturbation screening and the proteomics-based approach, such as co-immunoprecipitation or so on. And however, these 2 approaches face several challenges. And so for example, while the RNA interference screening can indeed discover some host proteins that are related to new virus production as an endpoint assay, after introducing the interference, it is hard to identify which stage does the infection in which the host vectors participates. And as for the proteomic-based approach, such as the co-immunoprecipitation, is able to identify the high affinity interaction between the viral protein and the host protein. However, it is very hard for them to identify the low affinity or transient interaction between the host vectors and the IAV proteins. Therefore, these 2 methods lack of spatial and temporal resolution. So therefore, to explore the virus-host interaction in a spatial temporal specific way, we have employed the proximity labeling assay, or the PL assay for short. This assay is based on the fusion of an engineered enzyme to a bait protein, which is a specific protein we're interested, or a subcellular localization protein, which can enter the enzyme to a specific space. And in our lab, we employ the TurboID biotin ligase for conducting the proximity labeling assay. And by fusing the TurboID with the bait protein at the appropriate location, we can perform biotinylation at the region of interest. And as the viral component assembling and budding take place at the plasma membrane, we fuse the TurboID with the bait protein PHPLCD1. PHPLCD1 is a domain on phospholipase C delta 1, also known as a black chain homology domain, or PHPLCD1 for short. It binds to PIP on the cell plasma membrane and therefore localized at the inner side of the cell plasma membrane. And by fusing the TurboID with PHPLCD1 domain, we are able to anchor fusion protein on the inner side of cell plasma membrane and perform proximity labeling to label a protein that appears in its 10 nanometer neighborhood with biotin. And since there is no endogenous biotin in the mammalian cell, we are able to control the labeling time point by controlling the addition of exogenous biotin. After adding a belting, the turbo ID can label the protein nearby the cell plasma membrane. However, to prevent the false positive hits in the results, we also employ two different kinds of stable cell line, which is the GFP turbo ID protein, expressing stable cell line, which the fusion protein is not anchored onto the inner side of the cell plasma membrane, and therefore can roam inside the whole cell and label this protein within the whole cell. The third one we use is the one where we truncated the enzymatic site of the turbo ID and therefore destroy the enzymatic activity of the turbo ID. So therefore, this stable cell line may not able to label any kind of protein. So to provide information about what effect does the overexpression of turbo ID or the background non-specific binding proteins are in the datasets. And after the labeling, the belting labeled protein were purified using the Stradivari beads and analyzed by mass spectrometry. So to ensure the subcellular localization of the fusion protein, we use confocal image to make sure that the localization of turbo ID. And as the orthogonal view shows, the turbo ID is indeed anchored on the inner side of the cell plasma membrane in both PHPLCD1 expressing cell line. So they are enriched at the inner side of the cell plasma membrane, with some of them in the cytoplasm. And for the one we deleted the PHPLCD1 domain, the GFP turbo ID fusion protein is in the whole cell. However, in the turbo ID signal, there is one region that has no signal, and this is the nucleus region. And we suspected that it is because of the GFP containing the nucleus localization sequence, so that it will recruit the fusion protein into the nucleus without the signal right here. So after that, to make sure the localization of fusion protein and biotinylation will not change upon IAV infection, we mock or IAV infected the stable cell lines before performing a PL assay. And the IFA result shows that a localization of fusion protein and biotinylation remain consistent. And as we expected, the fusion protein in PHPLCD1 GFP turbo ID cell line stayed on the cell plasma membrane and is not affected by IAV infection. And the localization of biotinylation stays at the side of the inner side of the cell plasma membrane. We also look into another two stable cell line and found out that indeed the IAV infection will not affect the biotinylation localization or the fusion protein localization. And so here we also check the one that the turbo ID was truncated, and we can see that the biotinylation signal is very weak. So it's the background it can serve as a background of the study. And after confirming that the IAV infection will not change the localization of fusion protein or biotinylation, we verified that IAV infection will not alter the expression level, the molecular weight, and the biotinylation profile of the fusion protein. And the wild-type A549 cell was here for the biotinylation background reference. And as the result shows, the two stable cell line expressing the full length of turbo ID does indeed show the biotinylation, and it is not in fact affected by the IAV infection. And on the right-hand side, the A549 wild-type cell and the truncated turbo ID expressing cell does not show any biotinylation pattern. And later, we confirmed that the efficiency of the elution method we chose to use. And as a result, the first elution, which is the E1 length, does retrieve most of the biotinylated protein, with there some residues in the E2 lane and the beads, and some of them left on the beads. And on the right-hand side, you can see that after the truncation, even though there is some weak biotin signal on the input and flow-through lens, the E1 and E2 does not show any biotinylated proteins. And after confirming the reliability of our elution method, we applied this approach to our mass spectrometry samples. And to further assess if IAV infection affected the expression level or enzymatic activity of the fusion protein, we performed Western blotting on samples collected at 5 different time points, which is the mock time, and we use it as a 0 hours post-infection time point in the study, and 6, 8, 12, 16 hours post-infection. And there's no significant change were observed in either biotinylation activity or fusion protein expression across different time points. And in the mass spectrometry result, we identified a total of 6,670 proteins from 3 different stable cell lines across 5 different infection time points. And in those proteins, there are 6,478 proteins are identified within the PHPLCD1 GFP turbo ID datasets, and 1,400 67 are in the truncated group. The rest of them are in the GFP turbo ID group. And so there are, in total, there are 4,652 proteins exclusively appeared in the PHPL-CD1 GFP turbo ID datasets. And so, however, when we try to filter the mass spectrometry data by deleting the proteins that that appears more than one dataset, which is those that also overlap with other protein dataset, other cell line datasets, we found out that this deletion will actually remove more than 60% of non-host factors in IAV lifecycle. And as we haven't come up with a better solution to perform data filtering using these three datasets, we decided to just proceed the downstream analysis from now on and to find out other way to filter later. And so in all the 6,478 proteins that we identified in the PHPL-CD1 GFP turbo ID group, we filtered out the proteins that only appears in one time point to exclude the effect that were caused by the single peak because it only appears in one time point, there will be a protruding peak that may cause artifacts. And so the remaining 6,409 proteins were grouped according to the abundance profile across the time points. And as a result, we are able to group those proteins into 24 clusters. And in all the 24 clusters, cluster 1 caught our attention. And when we take a closer look to cluster 1, the abundance level of cluster 1 protein started to increase since 8 hours post-infection. And since 8 hours post-infection is considered the onset time point of assembling a budding process, we suspected that the proteins in cluster 1 may have assisted the assembling and budding process for its abundance level in the vicinity of cell plasma membrane started to increase at the 8 hours post-infection. So therefore, we submitted the 202 proteins within cluster 1 to String database to investigate their protein-protein interaction. And as a result show, most of the protein falls into this big cluster. However, this big cluster does not have any functional annotation, so it provides us limited data. We then subsequently submitted the cluster 1 protein to gene ontology and reactome analysis. And in the cell component part, most cluster 1 protein were labeled as cytosolic proteins with some membrane-related organelle in the results. And as for the biological process, there are several protein transportation-related processes, and for the molecular function group, most of them are protein binding-related function. As for the reactome pathway analysis, there are multiple immuno-related pathway, which is interferon signaling and HIV-infected related pathway. Though there was no IAV-related pathway, the membrane trafficking pathway and the ER-Golgi-related pathway draw our attention and will be discussed later. And even though we're not able to directly filter the PHPL-CD1 GFP turbo ID datasets with the other two stable cell line datasets like we originally intended to, we still want to compare the gene ontology and the reactome pathway result across all these three cell lines. And there are only 142 gene ontology results are overlapped within the whole three group. And so, with 2,950 that uniquely appears in the PHPL-CD1 GFP turbo ID datasets, indicating that these three cell lines did indeed label different populations of proteins. And in the GFP turbo ID result, there are mostly mRNA biogenesis or RNA-related pathway that is probably caused by the nuclear localization sequence we previously mentioned that is included in the fusion protein. And the cellular component and the molecular function are also very distinct from the PHPL-CD1 GFP turbo ID results and mostly focus on RNA-related pathway or nucleus-focused subcellular components. As for the PHPL-CD1 GFP truncated turbo ID group, in the result they are also very different from the other results, cell line results. And these results indicating that our original strategy of trying to use these two datasets to eliminate the false positive hits might work, though we do need a more delicate application instead of just directly deleting everything that appears more than 2 times. And as I mentioned earlier, the membrane trafficking and the ER-Golgi transportation pathway draw our attention. So for IAV, we rely on vesicle transportation system to recruit some of the viral components to the cell plasma membrane for the further assembling and budding process. Therefore, we decided to look into one of the protein candidates in cluster 1, which is RIP1A. RIP1A is a protein that belongs to the RAS-related protein family, and they are a group of small GTPase or GTP binding protein that belongs to the RAS superfamily. They are able to bind or hydrolyze the GTP and to control the vesicle fusion and transportation in the cell through their GTP and GTP cycle. And RIP1A is the one that participated in the pathway where the protein transported from ER to the Golgi. And since we suspected that RIP1A might facilitate IAV assembling and bonding process, we speculated that RIP1A's subcellular localization might change during infection since the abundance level of RIP1A in the vicinity of plasma membrane has changed. And to examine whether RIP1A's subcellular localization changed upon IAV infection, we infected A549 cell and subjected them to IFA. And in the mock-infected cell, the RIP1A are mostly focused on the perinuclear region, while after infection it outspread to the cytoplasma, and after 4 hours at 4 hours for the infection, the outspread started and remained until the end of the assay, which is the 16 hours post-infection. And so to take a closer look at when did this phenomenon happen, we applied live cell imaging, and in fact the mCherry RIP1A expressing A549 cell with the PRA-based GFP NS1 expressing reported virus. And as a result, the outspread started to show around 3 to 4 hours post-infection and remained on this phenomenon till the end of the imaging, which is 18.5 hours. And so to determine whether RIP1A has a role in IAV life cycle, we assess the impact of RIP1A depletion on the production of progeny virions using the RNA interference approach. An A549 cell was transfected with either RIP1A targeted siRNA or non-targeting siRNA, which is called scramble in this research, and infected with IAV. And at 24 hours post-infection, both infected cells and the cell culture medium, which is the culture supernatant, were collected for further analysis. And in the cell part, the cell was submitted to 2 different assays, which is the Western blot analysis and RNA extraction. For Western blot analysis part, we use Western to make sure that RIP1A knockdown has successful and the IAV infection is not affected by the siRNA transfection part and the cell viability. For RNA extraction part, we use it to investigate whether the viral genome replication will be affected by the RIP1A depletion. And for the cell culture supernatant part, we submitted them to 3 different assays, which is the PLEC assay, the HA assay, and RNA extraction assay. And for PLEC assay, it is used to investigate the amount of infection progeny virion, and then the HA assay is to quantify the total viral particle, whether it is infectious or not. And for RNA extraction, it will give us a view of gene contact of the released virions. So the Western blot result indicated that RIP1A is indeed knocked down and the IAV infected is confirmed. And then we look into the PLEC assay result, which is the infectious progeny virion. We observed a significant reduction in the number of infection virion produced by RIP1A depleted cell compared to the scramble controlled ones. And this result led us to think that RIP1A depletion maybe impairs the efficiency or the robustness of the virus assembling process, potentially increase the proportion of the defective interfering particles that lacks a full set of viral genomes. So to test this hypothesis, we use the HA assay to quantify the total number of particles that are produced under RIP1A depletion or N control, and calculated the ratio between the infectious progeny virions to the total produced viral particles to assess the assembling efficiency. And the HA result shows that the RIP1A depletion led to an about 2-fold reduction in total viral particle compared to the control. So to combine these 2 results, we calculated the infectious versus total viral particle ratio. However, the ratio didn't exhibit significant change. These findings suggested that RIP1A depletion does not significantly alter the proportion of the infectious to total particles, but instead reduce the overall production of viral particles. To further explore whether RIP1A depletion affects the viral replication process, we quantified the MPVRNA in both intracellularly and in the culture supernatant. and the NP-RNA level in the supernatant showed a slightly changed but not significantly, statistically not significant decrease. And with intracellular NP-RNA level were significantly reduced in the REB1A depleted cell, this result implies that REB1A may contribute to efficient viral genome replication, although the precise mechanism by which REB1A facilitates this process needs to be further investigated. And so to further investigate whether IAV protein interact with REB1A, we use IFA to examine whether several IAV proteins were co-localized with REB1A during the IAV infection. In the previous study, the co-IP result indicated that REB1A might potentially interact with M1, M2, and NA. So therefore, we decided to take a look at these proteins. And as a result show, REB1A does not show co-localization with REB1A. However, in the linear profile of the region where REB1A aggregates, it seems like REB1A and M1 protein signals somehow, so the peaks are somehow exclusive to each other, so it's not co-localized at all. And as for the M2 protein, it seems like there's no co-localization at all, as M2 protein mainly appears in the cell plasma membrane, while the REB1A mostly stay at the paranuclear region, and some of them are outspread to the cytoplasm during the IAV infection. However, to see whether if M2 and REB1A were co-localized at the cell plasma membrane, we took a closer look at the region. However, it seems like there's no significant co-localization at the plasma membrane either. Last but not least, we took a look at the NA protein, and while NA was scattered within the cytoplasm, REB1A did seem to co-localize with NA in some region, which is here. So we take a closer look to this region, and as a result shows, the linear profile did not show strong co-localization between the NA and REB1A protein. To further assess whether REB1A physically will interact with IAV protein or not, we performed immunoprecipitation of endogenous REB1A with IAV infected A549 cell. However, the results show that no detectable immunoprecipitation result of IAV protein with REB1A protein. So therefore, further investigation is needed to determine the precise role of REB1A during IAV infection. In summary, we identified more than 6,000 proteins in our PL proteomics results, and among them, 6,409 were submitted to further hierarchical clustering and grouped into 24 clusters. And from the 24 clusters, the cluster 1 was selected for its abundant profile caught our attention. And REB1A was selected from the cluster 1 for its function and was for its ER-Golgi transportation function. And it showed distinct cell subcellular localization upon IAV infection. And REB1A depleted cell will produce less infectious progeny virion and total viral particles, but the ratio remains unchanged. The amount of intracellular NP-VRNA in REB1A depleted cells were lesser than the control, while the supernatant only slightly decreased and without significance. REB1A did not co-localization with the M1, M2, or NA protein, and did not show detectable interaction physically with all IAV proteins. So, as the discussion part, for the data filtering part, we haven't developed a suitable system to filter the PL proteomic datasets with the GFP turbo ID cell line and the PHPL CD1 GFP truncated turbo ID datasets. But we are considering whether a scoring system that includes these two datasets will be a reasonable solution. And also, while we are doing the clustering, we deleted the protein that only appears in one time point to prevent the disruption of protruding peaks. But these proteins may have partake in IAV life cycle in a transient way. And we included all the proteins regardless of their statistically significance, which may cause problem after the abundance level was standardized because the small fluctuation of the abundance may change the pattern and cause a lot of problems while we're doing the clustering. And as for the REB1A part, from the lack of interaction and co-localization between IAV proteins and REB1A protein in both IFA and IP results, we suspected that REB1A might not affect IAV life cycle with the direct interaction, but in an indirective way, such as they will somehow disturb the whole cell mechanism. So. And also, while a data set of REB1A depleted cell is the first complete data set we can generate, which is including all the 5 assays I just mentioned, the plaque assay result, the HA assay result, the RNA extraction in both cell and in the culture supernatant and the Western blot. We actually has a lot of different results previously in our lab, which is shown on here. So as we can see right here, although the infectious progeny variance was still reduced, this one is the same. However, when we take a look at the VRNA amount, we can see that in the MOI1 results, the REB1A depletion cell actually caused more VRNA amount. But in our study, although it shows like smaller standard deviation between technical replicas, we still need to take a closer look or to repeat it. And we suspected that the fluctuation of the result may be something caused by the REB1A knockdown is not stable or by the transfection process. So in the future, while this study allowed us to explore dynamic influenza host interactome in the vicinity of plasma membrane, there are more investigation to do. For example, we should develop an adjustable scoring system that including multiple criteria to better evaluate the protein candidates in a more comprehensive way instead of a simple deletion. And second, since we're not sure how REB1A depletion affected IAV life cycle and is suspected that it has something to do with ER-Golgi disturbance, we can investigate if ER or Golgi are ruptured during IAV infection and see if the damage colocalized with the REB1A with the markers. And third, while we examine how REB1A depletion affected virion production, which has happened in the late stage, however, the REB1A will show different localization at a rather early stage, which is around 3 to 4 hours post-infection. Therefore, it is worth examining earlier time point, which is aligned with the REB1A pattern change. So we can probably look into the events such as entry or virion uncoating or nuclear imports. And so also, because REB1A knockdown results shows a bit different the fluctuation, we should develop a REB1A knockout cell line that to further stabilize the REB1A removal and to also remove the influence caused by the transfection process. And so we do believe that this REB1A knockout cell will provide us with more reliable results. And also, while PL's proteomics can indeed provide us information about the the proteins that transiently appeared in the cell plasma membrane, it may have some of the proteins might just appear there and have nothing to do with IAV life cycle. So we should probably take a look at other plasma membrane focusing approaches such as cell fractionation by intracellular polymerization to further get more information about host proteins that may partake in IAV life cycles. So this is the end of my presentation, and I want to thank Dr. Wang for supervising me along my master thesis. And I would like to thank everyone here today and also the committee members who are here to give me suggestions, and thank you. This is my end of presentation, and I appreciate any question or suggestion. Thank you.